Hi, I'm Professor Johnson, and in this video we are going to be talking about the sex chromosomes and sex-linked genes. So I'll start with a story. This is a story about a scientist known as Thomas Hunt Morgan. He was a scientist in the early 1900s who was studying genetics, and he was using fruit flies as his organism of choice to study this. Well, one day in the lab, Thomas Hunt Morgan, or one of his researchers in this lab, made an interesting discovery. So they were crossing these red-eyed fruit flies with these white-eyed fruit flies. And when they did that, they always ended up with red-eyed fruit flies. So you would probably come to the assumption that the red-eye gene is dominant in this case. They then would perform a second cross with the F1 generation, much like Mendel would. And when they did this, they came to the normal 3-to-1 ratio that you would expect. However, there was something peculiar. When they went to determine, or when they went to look at the sex distribution in this F2 generation, they noticed that all the females had red eyes, while half the males were white eyes. And this was really kind of a peculiar observation. They thought maybe that for some reason the female fruit flies with white eyes weren't viable for some reason. They were dying off, so they didn't end up with those. So they actually really wanted to test this with another experiment. So they took the F1 females, which were red-eyed and heterozygous for the trait, and crossed them with their dad, or one of his brothers, to really perform a test cross to determine what this is. Well, when they did this, they found that you could get red-eyed females and white-eyed females, and you get red-eyed males and white-eyed females. So this is what you would expect to get in this experiment. So what was going on in this? They finally came to the realization that, oh, these genes were sex-linked. So what does that mean? That means that the trait was on one of the sex chromosomes. Normally when we look at sex-linked genes, the sex chromosome where it's going to be on is the X chromosome. And so you can see that I'm showing the chromosomes here as X and Y's to delineate what they are. And then the gene that we're interested in is the gene for eye color, with red being dominant to white. So I'm showing the big R here and a little r here for red eyes and white eyes. So you do the Punnett square and you can see, sure enough, the red-eyed females, or the red-eyed flies always would be females in this F2 generation, while the white-eyed females, or the white-eyed flies, would always be males. So as you can see, there's genes on these chromosomes, and it's really how these genes are inherited then that determines what the organism will be, either red-eyed or white-eyed in this case. Alright, so now I want to talk a little bit about aneuploidy. So aneuploidy is when you have an abnormal number of chromosomes. So this is what a normal karyotype would look like if you would isolate all the chromosomes in your cell, line them up. This is the 1 chromosome, and you go all the way down to 22. This person's a male. This is the X chromosome, and this is the Y chromosome. They're not X and Y shaped, but the X chromosome is significantly bigger than the Y chromosome. So the X chromosome has a bunch of extra genes on it. Well, it is possible to have the wrong number of chromosomes. So this is due to non-disjunction events that can occur during meiosis. And then when you have fertilization, if one of the numbers of chromosomes are wrong, you can end up with an extra set of chromosomes. Usually this is a lethal situation. So most of these larger chromosomes, especially, if you end up with missing one or having an extra one, so being either monosomy or trisomy, you end up dying. It's a lethal situation. Usually these individuals don't even survive to birth, but if they do survive to birth, it's generally a short-lived life. Now there are a few exceptions to this. So trisomy 21, commonly known as Down syndrome, is a situation where someone with an odd normal number of chromosomes can actually live and function in society for a large, a significant portion of time. So they're a lot of these people can grow to be adults. However, if you know someone with Down syndrome, you know that they live very different lives from the rest of us. They have reduced mental capacities, which don't allow them to develop quite as much as us. And that's really the best case scenario with having too much or too little of any one of these 22 autosomes. Let's look at what happens when we have odd numbers of the sex chromosomes, however. So I show you it here. This is a female that had an egg with a non-disjunction event. So you have the two X's over here. This individual didn't get any of the sex chromosomes. They only have the autosomes in this gamete, and this gamete has both of the X chromosomes. This is a normal male that's going to be donating an X and a Y chromosome. So you do the Punnett square, you can see that you end up with 4 different situations. A triple X situation, one where there's 2 X's and a Y, one where there's only a Y, and one where there's only an X chromosome. So the 3 X's, we call this situation triple X chromosome. And for the most part, these individuals always end up being female, and they develop fairly normally. They just have this extra X chromosome. Now you may say, well, maybe there's just not a lot of genetic information on the X chromosome. The X chromosome is actually relatively large, and there is going to be a lot of genetic information that's coming from it. So you may, although there's not much wrong with individuals that have an extra X chromosome, you might have suspected that there is. Similarly with the X and Y chromosome, this is known as Klinefelter's syndrome. These individuals develop as males, but they do have slightly more feminine characteristics, but otherwise can pass in society as fairly normal individuals. This individual that's missing an X chromosome would develop Turner syndrome. There are some functional characteristics and slightly reduced mental capabilities in individuals with Turner syndrome, but for the most part, they develop into a functioning individual, where you wouldn't expect that with someone that has a large chromosome missing, because for the most part it was autosomal. Now the person that got only this Y, they actually wouldn't survive to birth. This is a non-viable combination here, just having a Y chromosome. So the X chromosome is important, but for some reason it doesn't appear that having too much of an X, or in these top 2 cases, or missing an X, is going to cause any grave problems. So what's going on here? Well, when you have X chromosomes, one of them actually is going to get deactivated. Now this is going to occur in all of your cells, but it's going to be random, and it's going to be fairly early on in development. So there will be some cells, I'm drawing this little dot here to represent what's really going on. So this X chromosome stays activated, this other one gets deactivated, becoming what's known as a bar body. It's named after the person that discovered it. So these genes on this X chromosome wouldn't get expressed, but the genes on this X chromosome would. In other cells, you might have this bar body, or this chromosome, get deactivated into a bar body, and this one stay active. So this is actually what's going on with calico cats. So if you know someone that has a calico cat, more than likely they're going to be a female. There is an exception to this, and that would be cats that would have this Klinefelter's syndrome to it. But for the most part, some of these cells early on in development had the 1X deactivated, and so they end up with these black spots on them. And other cells early on in development would have had the black X instead of the orange X deactivated, and so would have these orange spots on them. And so they ended up with these two different colors on their different furs, because all the cells subsequent from that would develop into a cell that was only expressing that chromosome. So part of the reason why having or missing an X chromosome isn't lethal is because if you have the extra X chromosomes, they're actually going to get deactivated. And if you're missing one, it does play some roles early on, and that's why you probably see some of these individuals with Turner syndrome actually have a notable phenotype. But for the most part, later development won't depend on having this second X, and so this 1X here is sufficient. So most of what's going on with the Y chromosome is significantly smaller, has less genetic information on it, so losing it or having an extra one isn't going to cause nearly as much problems as it will for the X chromosomes, the autosomes. All right, I hope that was informative, and I'll see you guys later.